Hey biology! Alright, so today we're going to be tackling DNA replication. Now this lines up with section 13.3 in your textbook, so if you wanted to read a little bit more about it, you can do so there. Today's learning objectives are two things. One, to explain the role of DNA polymerase in copying DNA. And then the second one is to compare DNA replication in prokaryotic and eukaryotic cells. So let's start off by doing a review of DNA structure. We're going to use this model here, this diagram, to identify what some of the important parts of the DNA molecule are. You can also review the video that I put together last week if you want a refresher on DNA structure. So first of all, this entire molecule right here is two strands that are connected together and then twisted around each other. So we refer to that as a double helix. The double part is because there are two strands, and then helix means to twist. So both strands are twisted around each other. Now, on the outside of the molecule, this structure right here that sort of looks like a ribbon here in this model is called the sugar phosphate backbone. And everything that's on the interior that's color-coded, so you can see that there's a green structure that's combined with a yellow structure, and every place that's always the case. And then you also see purple combined with red, and those are going to be the nitrogenous bases. Now, this model here doesn't really identify each individual component as well as this model does. You may remember this from the previous video here. So they're showing the sugar phosphate backbone as simply one continuous piece, but in reality, the sugar phosphate backbone is a repeating sequence of sugars and phosphates and sugars and phosphates, and so on. And then, of course, the bases on the inside, you'll notice that the green corresponds to the green adenine, and the yellow corresponds to the yellow thymine, the purple corresponds to the purple cytosine, and then the red corresponds to the red guanine. So every place where there's a cytosine, there's a guanine. Every place where there's a thymine, there's an adenine. All right, let's go ahead and take a look at how DNA replication proceeds. First of all, what we're looking at right here, this entire section here is referred to as a replication bubble. And that's because as the molecule is copied, it's actually opening up into little bubble-like structures. So this is where our first enzyme here is going to be involved, and this is called DNA polymerase. Now, if we break down that term DNA polymerase, it makes it really easy to tell what the job of this enzyme is. So DNA tells you that it's going to be building DNA molecules, and then we know what poly means, that's many, and mer means pieces. So this is an enzyme that constructs DNA molecules out of many pieces. Now you'll notice that there are two DNA polymerase enzymes here, and what they're essentially doing is using the original strand of DNA as a template to base pair new DNA nucleotides. So if this molecule was to be opened up, then what happens is we have bases here that are no longer paired with their complement. So for example, here's a thymine right here, this yellow thymine. There's another thymine, there's an adenine, there's a thymine. Way down here, there's an adenine and a cytosine and a guanine, and they're not paired up with their complements. So what DNA polymerase does is that pairing process. It actually constructs the new molecules by pairing up complementary nucleotides using the original DNA as a template. So this DNA polymerase is moving in this direction, that would be down on your screen, and this DNA polymerase on the right-hand side is moving up on your screen. This area right here we call a replication fork because this is where the two strands split. You can see that up here at the top is the original DNA that hasn't been copied yet, and then everything that's going on inside of the bubble, that's where the replication is happening. Now, if I proceed down here to the bottom of the image, you'll see that there's another replication fork, and below it is some DNA that has yet to be copied. So where new nucleotides are being added is exactly where the enzyme is. All right, so the next part of this that's really important to note is that each of these strands has been color-coded for a reason, and that's because the blue strand here, if you notice at the top of the model where the DNA hasn't been replicated yet, or the bottom of the model where the DNA hasn't been replicated yet, if you sort of follow along either one of these two strands, so as I follow it along here it goes, and it sort of twists around the new DNA that's being replicated. I could do the same thing with the other side, and I can see the blue here. It twists around the new strand that's being replicated. The blue strand represents the original DNA. And the orange strand on the inside here, and on the inside here, that represents the new DNA. So what I'm going to end up with is two molecules of DNA, and each of those molecules is going to be made up of one original DNA that was used as a template to make the one new piece of DNA. So because each DNA molecule is half old DNA and half new DNA, we refer to DNA replication as being semi-conservative, because to conserve something means to save it. So in looking at this model here, you can see that the blue strand here was saved. Over here, the blue strand here was saved, and then the orange strand is new. So in this side here, half of the DNA molecule was saved and is original, and in this over here, half of the DNA was saved and is original. Now let's take a look at prokaryotic DNA replication. Remember, prokaryotes are bacteria. So in this example here, we're seeing that prokaryotic DNA is a circular molecule. So if I was to start here, I can move around and I can see that there's a DNA in a circle here. Here's one of those replication bubbles. So if I were to label the parts here, here's a replication fork here. There's a replication fork right there, just like we already saw. And this DNA here that's color-coded in orange, just like in the last illustration, is the new DNA. And all of this stuff out here is original DNA that hasn't been replicated yet. If you look closely, you'll see that these two strands here, the one here and then the one down here on the bottom, each of those two strands is half blue, old DNA, and half orange, new DNA. So it's still semi-conservative replication. Now, how does this compare to eukaryotic DNA? It's similar in a few ways. One is that there are replication forks. Two is that the molecules that are created are half old DNA and half new DNA. But here's where some differences show up. Eukaryotic DNA is not circular, it's linear, so there are ends to it. There's an end up here. If it continued on, we'd find the end. There's an end down here. If it continued on, we'd find the end. And unlike prokaryotic DNA, where we saw a single replication bubble, there are actually multiple replication bubbles. Here's one right here, and if we zoomed in on this, it would look something like this illustration here that we saw earlier. Here's another replication bubble, and if we zoomed in on this, it would look something like this, exactly like what we saw earlier. So that's a really simple view as to how DNA is replicated. Now, in reality, there are many, many, many enzymes that are involved, and this process is a lot more detailed. But what we need to take away from this is how DNA is replicated in a semi-conservative way. That was objective number one. And how eukaryotic and prokaryotic DNA replication compare, and that was objective number two. All right, I think that pretty much takes care of it for us this time. In our next video, we're going to look at what DNA is actually used for. All right, until then, take care.